Okay, guys, we'll get started. So I switched over to OneNote. I know a lot of you guys like using those note-taking apps. It's probably easier for me to do stuff with this note-taking app. So I switched to OneNote. It's still all a PowerPoint. It's all the same. But I can like zoom in and zoom out on PowerPoints. And when I'm drawing, it's a lot easier now. So we'll use OneNote. So anyway, today we're going to start neurophysiology, start learning about how our nervous system works, how it all kind of works together. We're going to go over the structures of the brain, go over the cells of the brain, how we sense our environment, and then go about our senses. Okay. This is going crazy now. Why is that there? Okay. Okay. So the intro to our nervous system. It'll work eventually. Why is that there? Okay. So obviously our nervous system, unconscious control, which is obviously very, very important. Let me just lower that. Has neurons, and any type of support cell is just called the glia. There are different types. Glia is just like an umbrella term that describes all of the cells within the nervous system. Okay. There's 10 to 11 million neurons, whatever that number is. That's a lot. Here's the very, very important part. There's regional specialization. We've mapped out the brain. We know what parts of the brain are responsible for what functions. And because of that, we have predictable symptoms within their nervous system that we can diagnose from. So we've mapped it out. We know if you start having issues with, if you're having auditory hallucinations, we know that the hearing structure is in the temporal part of your brain. So if you're having auditory hallucinations, we'll know to look at the temporal lobes. If you're having visual hallucinations, we know that the primary visual cortex is in the posterior part of the brain. So if you're having visual disturbances, you may have a problem with the posterior part of the brain. The whole thing is mapped out. We know exactly where things are. This should be reviewed from anatomy, all the different anatomical terms. I'm not going to test you in physio about anatomical terms, but you should know what they mean so that if I describe them in a question, you'll know what it is. But again, rostral and caudal, head and tail, dorsal and ventral. Dorsum is the back, ventrum is the front. Think of a dorsal fin on a dolphin or a shark, it's on the back. Anterior, posterior, it does change, anterior and posterior, when it comes from the brain. When you get to the spinal cord, everything kind of flips. Okay. Again, medial and lateral, which way they're going. Ipsilateral, again, a lot of this stuff in the beginning is just definitions, so we all know what they mean before we go on to the other stuff. Ipsilateral is 2 points on the same side. So right arm, right leg is ipsilateral. Contralateral means the opposite side. So 2 points on opposite sides. If you have a stroke and you have a motor stroke, we'll get to this in a little bit, the left part of the brain sends the signals to the right side of the body. So it's a contralateral change with the innervation there. This seems to be going slower than the PowerPoint was. Okay. And still, you have those coronal slices, horizontal planes, all those things you learn in anatomy. We're just going to glance over it, just so you know that they're there. Here's what I was talking about before. So above the brain, you have the rostral, caudal is the posterior, you have dorsal, and you have ventral. In the brain, the dorsum is the top part of the brain. The underside is the ventral Anterior is rostral, and the posterior is caudal. If you look below the brain, it all switches. It turns 90 degrees. And so I have this blank one here. You'll see blank ones, this is because I draw stuff in. So if we have, this is like a huge spinal cord. We have different parts of it. Anyway, spinal cord. Here's our dorsal, which is the back. Here's the ventral, and so forth. So it changes when you get to the spinal cord. Just remember that. In the brain, the dorsum is the top of the brain. In the spine, the dorsal part is the back. So it switches. The anatomical terms switch when you get to the spinal cord. So don't get confused in anatomy when you get to the spinal cord, because the dorsal part of the spinal cord is the back of it. The dorsal part of your brain is the top. So it moves. Okay. Let's talk about nerves and our prototypical neuron. If this is really annoying for you guys, let me know. I'll go back to PowerPoint. Okay. So we have the soma, which is the cell body that has the nucleus. This part of the neuron is demyelinated or unmyelinated. There's no myelin in the cell body. That's going to come up in a few minutes as being important as far as why. Cell body has no myelin. We have the neurites, and these are extending from the soma. These are these little branches right there. We have the axon, which is the whole length of the neuron. This is where all the action potentials are propagated and they get sent down the row. So the axon is where it all goes down. And we have dendrites, and they're the things that receive, these are little things there on the outside, they receive the impulse from another neuron, and that's how we get transduction of a signal. These area right here, this is the, you can call it the pre-synaptic. You can also call it a terminal Booten, another way it's called, but this is where the neurotransmitters are. And if at the presynaptic terminal, the terminal Booten, if this is where all the vesicles are, what voltage-gated channels are also right here as well? Not sodium, calcium. Remember, calcium is right at the end there because that's needed for exocytosis to get all that neurotransmitter to be released. So that's where the calcium channels are, right at the end where that terminal Booten is. So this is your prototypical neuron. Here are just some definitions. You'll see things called gray matter on a CT scan or an imaging. Gray matter is the cell bodies in the CNS. It's gray because it doesn't have any myelin. What is myelin made up of? It's fat. What color is fat? White. It's white. If you go buy a steak, it's white. I mean, yes, we have adipose tissues, that's fat, but if you just go to the meat department and look at the steak, it's going to be marbled. All that white marbling is fat. So therefore, where is it? I think it's on the next one. So gray matter is the cell body. There's no myelin there. There's no white substance. Therefore, it's gray. So the gray matter is the cell bodies. We have the cortex, referring really to the cerebral cortex. A nucleus is a clearly defined mass of neurons. Again, these are just definitions. You're going to see words later on. They're called nuclei or tracts, but you have to know what they mean. A locus is a clearly defined group of neurons, but smaller than a nucleus. Substantia, well less defined group of neurons. We're going to talk about that with a substantia nigra when we get to the structures of the brain. A ganglia is just a collection of neurons in the peripheral nervous system. Okay, so ganglia only occur in the PNS, peripheral nervous system. We have, here's the white matter. Here we go. So white matter, These are the axons. They're white because they're aligned in fat, and that's why it's white matter. So whenever you see the words talking about the brain or the spinal cord, tissues, gray matter, cell bodies, white matter are the axons because they have the myelin. You have tracts, which are just a collection of axons all together. Just think of like internet cable or your phone wires. If you ever cut an internet cable or phone wire in half, there's like 6 to 10 wires inside of it. That's the same thing what a tract is. They're just going from one place to another. A capsule is just a group of axons connected to the cerebrum and the brainstem. A commissure is a crossover. Just think of crossover. So we have a white commissure in the brain, and that tells the signal to go from the right to the left side of the brain. They're called commissures. That's where they cross over. We have a lemniscus, which is a ribbon-like tract. And the last one is just a plain old nerve. So just, again, casually to make sure you know, don't sweat yourself going crazy over these definitions. Just understand just general what they mean. So our nervous system can be broken down into central and peripheral nervous system. The central is just your brain, spinal cord, and your brain and your spinal cord are covered in a layer of tissue called meninges. There are 3 layers, the dura, the arachnoid, and the pia mater. They are in order. Dura is the outermost layer. The pia mater is the innermost layer. You have to know these orders, okay? If you ever do a lumbar puncture, which AA you will be doing, PAs you might be doing, you have to know how far to go in because we want to get to the layer where the CSF is. Our CSF is located in the subarachnoid space, so it's under the arachnoid mater. And that's where you'll find all this CSF fluid. If you're not hitting that area, you won't get any CSF because it's not located there. So just remember DAP. Peripheral nervous system is everything outside of the spinal cord and the brain. So all your peripheral nerves, your spinal nerves, most of your cranial nerves, I know you guys started doing that a little bit today with anatomy. 2 of them, 1 and 2, just come out of the brain, but everything else comes out of the brainstem. Other definitions you got to know. And just like if you guys ever played a sport or you got a new hobby, the first week or 2, you're just trying to figure out all the words that go along with that sport or that hobby. And once you get used to it, then it's second nature and you just talk about it. Afferent nerves are sensory from the periphery to the CNS. So just think about if your brain and our nervous system is just one big computer, everything that you plug into that computer is an input. If you have, like I have a pen is considered an input. My screen is sensing what I'm doing to it, and it gives you a result. So afferent nerves carry sensory information. So if your phone goes off, you feel a vibration in your pocket, that is an afferent signal sending the vibration to your brain that you know you have a vibration going on in your leg. Efferent nerves carry from the CNS to the periphery. Okay, so that's efferent. Efferents are away. I know it sounds like you want the afferent to be A for away. It's not that way, it's the opposite. So afferent goes into the CNS, efferents leave the CNS. And we have our motor nerves. Obviously, they're going to be efferent because they're leaving the brain. So motor nerves are efferent nerves as well. And obviously, they're going to go to the skeletal muscles so we can move around and do things. So if you ever have an infection of the meninges, it's called meningitis. And if it's the infection of the actual brain tissue itself, it's called encephalitis. So meningitis is the meninges have irritation. Encephalitis means the brain tissue itself, the parenchyma of the tissue is actually what's causing or infected. So we can also divide the nervous system depending on its function. So we have the autonomic nervous system and the somatic nervous system. Soma just means body. So it's all the sensory and motor communication from the outside environment to the brain. Your soma, your body. Skeletal muscles and joints and so forth. The autonomic is where we're going to spend a lot of the time because that's the biggest effect that it has on our body. We have the sympathetic, fight or flight. Just always put those two together. Sympathetic nervous system, fight or flight. A bear's attacking you, what are you going to do? You need to do things. The sympathetic system will do them for you. Increase your heart rate, make you breathe deeper, puts glucose into the bloodstream so that you are ready to do whatever you got to do. Your eyes dilate so you can get a lot of light in, so you see what's going on. Those are all sympathetic things. Your parasympathetic, rest and digest. Okay, so right now you're all just sitting around. Your parasympathetic system is in control. You don't need to do anything. You're not scared. You're not trying to run away from anything. We're just resting and digesting, and that's your parasympathetic system. The last part of the autonomic is the enteric nervous system. This is that gut brain. We'll talk about this a lot more when we get to GI, but there is a lot of connections between what we eat and what effect it has on our brain. So let's keep moving. Okay, structures of the brain. We have the cerebrum. Okay, you also hear it known as the cerebral cortex. Okay, same thing. Input and controls movement of the contralateral side of the body. Okay, so remember, opposite side. So if it's the opposite side, what word did we just learn? What must be present somewhere for this to happen? Commissures. Okay, remember, we're crossing over. Any crossover is a commissure. So just remember that. If we're crossing over, we have to have a commissure somewhere. We have the cerebellum, which is behind the cerebrum, right in the back. This is very, very important for motor system, but think about muscle coordination. This is where the cerebellum comes into play. Okay, what happens if you're drunk and you get caught by the cops? What do they make you do? Walk in a straight line. These are field sobriety tests. These movements are coordinated movements to test your cerebellum, which gets deranged when you're drunk. Alcohol is toxic to the cerebellum, and that's why you have motor issues when you're drunk. Coordination problems. You can't walk in tandem. You can't walk on your heels, you're going to fall. That's what the cerebellum does. So balance, coordination, specialized movements. Just walking is not the cerebellum. But if I tell you to stop, turn around, that is a coordinated movement. So you see people who have like Alzheimer's or Parkinson's. Parkinson's affects dopamine, which is your cerebellum, in other parts. They can't do that. They're going to stop, and they're going to shuffle their feet like this to turn around, and then they'll go. So that's what the cerebellum does. And we have the brainstem. The brainstem is that area that comes out of the foramen magnum of the skull and goes down, obviously, to complete your spinal cord. Brainstem, vital body functions, heart rate control, breathing control are in the brainstem. So what does that mean from a clinical perspective? If you've had a stroke and you just have muscle problems, that means it was just the cerebral cortex. But if you have a massive stroke and now you can't control your own heart rate and your breathing, that means that you had an infarct in the brainstem, which is really bad because that controls our vital processes. Brainstem infarcts are most likely going to be deadly because they control our vital processes. You'll be on life support, but brainstem, that's where the brainstem is important. It's very rare for it to get infarcted, but it can happen, and you'll see it because your vital signs are messed up. They're not going to be able to control their vital processes. Okay, so here again, in our spinal cord, we have our meninges, arachnoid, and there's our dura mater. This is our spinal cord, so obviously we have to have spinal nerves that come off at each vertebrae. You have your cervical nerves, thoracic, your lumbar, and your sacral, and you'll learn them all as you go through anatomy. But Remember we said in the anatomical directions that when you go from the brain down the spinal cord, the orientation switches. So the dorsal is now in the back, the ventral is in the front. Every time you see a dorsal root, it's always going to be a sensory root. Dorsal is always going to be sensory, so it's always going to come in that way. The ventral root, which is the front part, is always going to be motor. So whenever you see these going forward, you ever see a spinal dorsal root? It's a sensory nerve root. The ventral is always motor, and then they combine together to make a spinal nerve. And they just go out to wherever it's got to go. Remember, you got to go from your spinal cord all the way out to your fingers. That takes a while. So you have to branch off from the certain levels. And you'll learn all that in anatomy. Okay? So just remember, dorsal is always sensory, ventral is always motor. So let's talk about our 4 brain structures now. Obviously, we have the cerebrum. We talked about that. That's our thoughts, perceptions, voluntary actions. So the frontal part of the cerebrum, your frontal lobe, is your personality, judgment. Yes, you can walk by and someone push you on the stairs. But your frontal lobe will tell you, no, don't do that. Okay? That's why kids are very immature, because that frontal lobe hasn't matured yet to tell them to stop, that don't do these things. Again, cortex interchangeable with cerebrum. It's all gray matter. And what is gray matter composed of? Not myelin, cell bodies. It's the axons. Oh, it's the cell body, the soma. Surface area of the brain is increased through folds and fissures. They're called gyri and sulci. So basically, you're increasing the surface area. If you're increasing the surface area, you got more nerves, and we're smarter. Koala bears. You ever hear the term like smooth brain? You call someone stupid, they're smooth brained? So koalas are smooth brained. And if you put a plate full of eucalyptus leaves in front of a koala bear, they will not eat it. Because it's not on a twig. They're not biting off a branch. They won't eat it. They won't recognize it as eucalyptus. That's what's smooth brained. So all these sulci of the brain, we've mapped them out. And we've labeled them into different lobes. They all have different purposes, so we'll go through those in a second here. Okay, Einstein. Einstein actually had an extra sulci in his brain. And actually, this brain was like stolen for a while. I think it was at the University of Philadelphia for a while, and then apparently it got stolen somehow. They got slices of it. But anyway, he has an extra sulci, so therefore, in the temporal region of the brain, the temporal region is responsible for abstract and spatial thought. So he had extra nerves, because he had more surface area in that area. Maybe that's what helped him come up with all those different things. Okay, so forebrain. It's your thalamus and your hypothalamus. That's it for the forebrain structures. Thalamus is a relay for all sensory pathways. All sensory input goes into the spinal cord, goes up to the brain, goes into the thalamus, and then it'll synapse to go to whatever it's got to go to in the brain. So the thalamus is a relay for all sensory. Some of it may be same side, some will be opposite. We'll get to that a lot later. And you can see here that cortical white matter. It's white because of the myelin. All the axons are there. Here's the corpus callosum, and that is the commissure. Okay, the link between hemispheres. We call it the corpus callosum. Same thing, it's a commissure, it's a crossing over. And in the gray matter, we have these deep cerebral nuclei. We have the caudate nucleus. You don't have to know all these things. Descending motor tracts go through here. Your basal ganglia, if you destroy that or have problems with that, you're going to have movement disorders. And here's your basal ganglia right here. You can see it's right near the hypothalamus. So the hypothalamus lies ventral. It's hypo to the thalamus. It's under, it's below. Command center for all your endocrine functions. So we'll deal with the hypothalamus a lot when we get to endo at the end. The hypothalamus is also a part of the structures that are outside of the blood-brain barrier. That comes in very helpful when it comes to certain problems and what's going to happen when we get to that in a second. Other sections in the forebrain, which we're going to get to at the end of this unit, towards the end, your amygdala, hippocampus, retina and optic nerve we'll get to next week, and our olfactory bulb, which is the sense of smell. Here's your meme. Gotta keep it PG. Okay, moving down, midbrain. What I want you to focus on here is this substantia nigra. Okay, substantia nigra. What does nigra mean? Black. Nigra is black. It's black because it's got dopamine in it. Dopamine gives it its black color. So, people who have Parkinson's patients don't have enough dopamine. What do you think happens to the color of the substantia nigra? It gets lightened, gets whiter. And you can only see that on autopsy. But that's how they figured out that dopamine was responsible for part of the problem, because they figured out the substantia nigra in these patients who had Parkinson's, that was gone. So substantia nigra is very, very important with regards to dopamine. And so this is substantia nigra, dopamine, we just talked about it. Don't worry about the symptoms. Hindbrain structures, we're going just deeper and deeper. We have the cerebellum, which is obviously in the back. We have the pons, and then the medulla, all parts of the brainstem as you keep going down. So the decussation of motor tracts occurs in the medullary pyramids. We have this structure called the pyramids in the lower cerebellum. In the medulla, not cerebellum, and that is where the crossover occurs. So remember, the left side of the brain controls the right side of the body. That's where it occurs. So that's what we're talking about there. Brainstem, obviously, conduit mediated tracts. Also have cranial nerves come out of here as well. So when you start learning about the cranial nerves, the origins, it's going to be the brainstem. And then here are your cranial nerves. Do you guys understand how mnemonics are? It's let me spell it. They're just ways to remember things. Whatever you decide works for you, who cares how silly and stupid it is? If it makes you remember things, keep it going. I know you have one nose and two eyes, so one is olfactory, two is eyes. Easy to remember that one. After that, you're on your own. But anyway, there's a bunch of these little mnemonics. I'll teach you one later on. We get to for reproductive. Point and shoot for males, very easy. Point, the erection is parasympathetic. That's what causes that. And the shoot, the ejaculation is sympathetic. Point and shoot. Then you remember it, and you remember it forever now. Stupid, however it works, it works. So just type in like mnemonics of whatever you're studying, and there'll be websites that all pop up. So I'm going to skip all this. We're going to go through these as we go through the different sensory issues and sensory, so we're going to go through these anyway, but I'm not going to ask you on a test which one is a glossopharyngeal nerve. That's an anatomy question. I may ask you, what does a glossopharyngeal nerve do? Or if, it also innervates the tongue with sensation of taste. So if I said, if your glossopharyngeal nerve is damaged, what part of the tongue will you lose taste sensation? That's a physiology question, okay? It's the back third, in case you're curious. The front 2/3 is your facial nerve. The back third is the glossopharyngeal. That's why people with strokes maybe lose different taste sensations. Okay, we're going to go through all these as we go through the sensations, so I'm not going to go through them now. Okay, so that was all the central nervous system. Now we're going to move on to the peripheral nervous system, which is just your spinal nerves. There's 31 pairs of spinal nerves, 12 pairs of cranial nerves. I said before, the optic nerve can be considered part of the central nervous system. I've seen these videos where they've done like petri dish experiments where they had nervous tissue and it essentially made brain tissue, but it grew eyes. Because it's brain tissue, the eyes grow out of the brain. So it grew eyes. I don't know why, but who knows? Except the vagus nerve, all other cranial nerves are just in the head and neck. Why is it called the vagus nerve? What is a vagabond? Anyone know what a vagabond is? It's a wanderer. So a vagabond, Vegas, wherever the root came from, but it's a wandering nerve. It starts in the brain, it goes all the way down to the abdomen. That's why it's called the vagus nerve. Okay, let's take a break here. Well, it's only 30 minutes. Okay, we'll go a little bit. This one's a quick little section. So, brain extracellular fluids. So, the volume of our brain is limited by the skull. If you twist an ankle or sprain your wrist and you get swelling, that skin can expand and allow all that fluid in that space. The brain has a skull over the entire thing, so the volume can't expand. If it starts expanding, you start getting intracranial pressure issues and a bunch of other problems. So the volume of the brain is kind of relatively fixed because of the skull. If the volume increases, you get risk of increased intracranial pressure, leading to decreased brain perfusion. That's why you don't want intracerebral pressure to go up. Reduces the perfusion of the brain. So we really need to make sure that the brain fluids are very tightly controlled. We don't want them to have a lot of waste and bad products to get in there. So we have a blood-brain barrier, which we'll discuss in a second. But there's three components. We have the plasma, blood plasma that floats through. We have our interstitial fluid, and that bathes our neural cells in the glia. And again, this is protected by the blood-brain barrier. We don't want a lot of things going into the brain. Pretty much just oxygen and glucose, maybe some other little things, and then out is the CO2, which is the waste product. And we also have our CSF fluid. They reside within the ventricles of the brain, kind of float around. They go all the way down the spinal cord, and they come right back up again. And this helps with free exchange of water and solutes between the brain and the CSF. So our ventricular system, this holds all the CSF within the brain, has choroid plexus epithelium. That is the cells that create our CSF. So they're constantly making CSF. And then it drains back into the venous system via the arachnoid granulations. So we're always making CSF, we're always reabsorbing CSF, so it's constantly being overwritten, I guess, what word is like exchanged. So our CSF is always being exchanged, we drain it out, we make more. As long as the balance is steady of what we're producing versus what we're able to get rid of, your CSF will remain stable and it's okay. Okay, but if it doesn't, then you start getting buildup of CSF, and that's that normal pressure hydrocephalus, if you've ever heard of that, NPH, just too much CSF volume on the brain. Functions of CSF, shock absorber. Okay, football players get concussions all the time. CSF is there, it's supposed to help with that, but the force of the impact is too strong, and it overpowers any type of absorption that CSF can do. Also helps to contain that or make a constant internal environment, and also removal of metabolites of the brain. So there's this hydrocephalus. Again, we have too much CSF on the brain, and there's different reasons for that. And there's your normal pressure. Again, you're not required to know the clinical correlations. They're just giving you real-world examples to kind of like reinforce the topic that we just talked about. Okay, so our blood-brain barrier. Okay, very, very, very important. So here's your typical capillary. Nothing's around it. Everything can leave freely as much as it wants. Here is a brain capillary. A couple of things there you see here. One, these astrocyte end foots. These are from the astrocytes, which is one of the cells of the brain we'll talk about in a few minutes. So they have these end foot processes. You may also see them as podocytes. But these provide a barrier around the entire capillary so nothing can get out. Those large molecules can't get through because it's so tight wrapped around the vessel, nothing can get out. Also, impermeable intracellular junctions, so again, nothing can get out. We have very, very, very tight junctions. Those large molecules can't go anywhere. So that's a part of the first part. So within the blood-brain, highly lipid solubles can go across. Remember, it's a phospholipid bilayer. They don't need any carriers. They can go right through. Oxygen, CO2, urea, nicotine goes right through, and alcohol goes right through. That's why nicotine, if you smoke cigarettes, you may get a feeling of euphoria when you do that because the nicotine goes in. Alcohol as well goes into the brain. Non-lipid soluble can't get into the brain. Glucose is the exception. Glucose is the only sugar your brain will use, so your body has to convert everything to glucose. So if you have those high fructose corn syrups and sucroses, those things have to be converted to glucose in the body so that your brain will use it. And glucose gets in because of those GLUT carriers. Glucose uptake is what GLUT stands for. I mentioned that a little bit yesterday. That's how sugar gets into the brain. So there are a few small areas that are outside the blood-brain barrier, and they're called circumventricular organs. These are outside the brain. Why do you think it's important that the brain knows what's going on in the body? Because if it's sealed off with this blood-brain barrier, it's not going to know what's going on, right? Some of the important things the brain wants to know about: pH of the blood, osmolarity, right? How many osmoles tell us if we're dehydrated or not, because that will be affected by the milliosmoles. The brain has to know these things to make changes with the hypothalamus. So we have a few organs and structures that are outside this blood-brain barrier, so the brain still has access to information that's going on in the rest of the body. The ones that they're called posterior pituitary gland, subfornical organs, median eminence, the pineal gland is outside, and this last one is this organum, don't worry about it, OVLT. Just remember that. You don't know everything that's in it. These are the structures that are outside the blood-brain barrier. We're only going to focus on a couple of them. Don't worry about memorizing all of them. The ones that I describe more, obviously, are the ones that are more important for the class. Again, exposed to solids in the blood. It's going to know what your glucose is like. How is your calcium? How's your magnesium, your sodium, your potassium? Brain's got to know these things. Some neuroendocrine control processes. When you see neuroendocrine, it means it's a nervous input causing an endocrine release of hormones. The posterior pituitary gland is innervated, has a nerve goes right to it to tell the posterior pituitary to secrete whatever it needs to. The anterior pituitary does not. So the anterior pituitary is just hormonal stuff through the blood. But the posterior pituitary, there's one other major gland that's innervated directly with nerves. Think about fight or flight. We want stuff to come out of real fast. What do you need in your bloodstream when you're fighting something or running away? Adrenaline. Okay, we call it epinephrine or norepinephrine. The same thing, noradrenaline, adrenaline. That comes out of the adrenal medulla, not the adrenal cortex, the inner part, and that is neurally mediated, which means a nerve goes right into that organ. If you are going through a sympathetic system or stimulation, a bear's coming out and you gotta run away, the nerve will tell the adrenal medulla to give us epinephrine and norepinephrine for us so that we can do whatever we have to do. So when you see neurally mediated, that means a neural input. There's a nerve that tells it what to do. The median eminence is in the hypothalamus. Again, we have to know what the hypothalamus is, a major gland as far as hormonal control. It's going to have to know what's going on in the bloodstream. And this OVLT is with fevers. And I think that's the next one. Yep. Nope, almost. So the reason why brain swelling is very bad. So brain or cerebral edema, increase in volume within the CNS. You have one called cytotoxic edema, and you're starved of O2. You have vasogenic, clinical signs, headache, again, the swelling of the brain. Again, don't go into detail with these things. Again, just reinforcing the idea about those things. And let's take a break now. Come back at 3:20. Okay. So moving on to cell neuroscience, all the different cells of the brain and what they do. Our brain function works because our cells can communicate with each other. One neuron, just by itself, can receive thousands of inputs from other neurons. So we have a lot of integration into the one neuron that gets to the brain, and then it can, whatever it's got to do in the brain. We do have some overlapping systems of classifications when it comes to the nervous system, because you can classify nerves by their size, you can classify by their shape, their function, so it kind of depends on what their job is. So these are the different types of neurites. We have unipolar, just a single neurite. You have bipolar, which has two neurites. We're going to talk about this when we get to the retina. We talk about sight and how we're able to see things. The majority of them are these multipolar ones, so they receive inputs from many different places, wherever else is intervening with that. Every time you learn something, you get a new connection. A new nerve touches another nerve, and you get a connection going on. So that's kind of like how we can define them that way. We can also describe them how they're organized. So we talked about before with those pyramidal neurons in the cerebral cortex that have all the motor in them. You also have these called stellate or star-shaped neurons that are there as well. So again, these are just describing things. So when you hear this word later on, a stellate neuron, you know it's a star-shaped neuron. You also can describe them based off of the length of the axon. How long is that axon? Golgi type 1s have very long axons. Golgi type 2 are very short. They kind of like help with like local circuitry. Also based off of their function. What are they doing? Are they a sensory neuron? Are they just there to receive sensory input from the environment? Are they a motor neuron? What's their job? Obviously to innervate your muscles so you can walk and do things. Or are they interneurons, which just go from one neuron to another neuron? So if you have, here's your cell body, there's your axon, there's the end of it, here's the next neuron, goes out, and then here's this one that goes to the muscle. This is the interneuron. It's between the neurons. That's the majority of the type we have in our body. So it's between, they only form connections with other neurons. That's why it's in the middle. And that is the interneuron. Okay, so those are the neurons. Now we're going to talk about the neurotransmitters in more detail. Okay, if it releases acetylcholine, it's a cholinergic neuron. So if it releases ACH, any neuron that releases acetylcholine is considered a cholinergic neuron. So here are the axons and presynaptic terminals. Remember, we have the soma right here. This part right here is called an axon hillock. And I think there we go. So this axon hillock, that's a very, very important area for control. We'll get to a little bit later on regarding inhibition. Inhibition of the nerves occurs at the axon hillock. And we'll explain why that's important later on. Then we have our axon proper, which is the myelinated part. We talked about the nodes of Ranvier yesterday and the saltatory conduction that goes the length of the axon to propagate and continue that exponential to go down the whole entire way. Then you get to the very end for the terminal boutins or the presynaptic terminal. What voltage gated channels are here? There's your calcium channels are there because that's where this exocytosis is going to have to happen. And that's where all the neurotransmitters are in those vesicles waiting for impulse, which is the calcium, to get it to release. Exoplasmic transport. How do we get stuff down these nerves? Okay, we have to transport stuff. You get oxygen, carbon dioxide, waste products, glucose. Nerves are living tissue. They need to be fed as well. So there's 3 different parts. This exoskeleton, don't worry about going crazy here. Just know that microtubules are like train tracks. And they just move stuff up and down that neuron. So they're just train tracks and they just stay there. There's anterograde transport, which is from the soma to the axon terminal. We have retrograde transport, where axon terminal towards the cell body now, so it's going backwards. And we have motor proteins as well, and those are the box cars that are on those train tracks that carry all that stuff along the way. So that's how we transport across the neurons, and this is where herpes comes into play. Herpes lives in the cell body of the neurons. During periods of stress, they come back out and they go all the way to the nerve endings, which are on your lips or wherever else you have herpes or polio, the other ones, rabies, tetanus. They live in the nerves. Because they live in the nerves, you can't treat them. That's why herpes is forever, because it lives in the nerves, not in anywhere else. Drugs we have right now can't penetrate the nerves, so we can't do anything about it. So they live there, they get in through the mouth, they travel down the axon into the cell body, and they wait there, and then when they're ready, they come back out, and that's herpes. Okay, neuroglia. There's 3 major types of cells in the brain we need to be worried about. The first one are the astrocytes. They provide scaffolding. They provide structure to all the nerves. They're also the cells that have the podocytes for the blood-brain barrier. So part of that blood-brain barrier. Those are the important things to know about astrocytes. If you ever heard of a brain tumor called an astrocytoma, it's dealing with these astrocytes. And we know what the astrocytes' function is for the blood-brain barrier. So what do you think is going to be compromised in someone with a astrocytoma? The blood-brain barrier, because there's no more podocytes. They all got destroyed from the cancer. Microglia is your immunity of the brain. That's the job of the microglia. They act like macrophages and they eat up smaller pathogens and they get rid of them. Remember, we don't want pathogens in the brain especially. The last one are oligodendrocytes. They maintain and produce myelin only in the CNS. That's the only place the oligodendrocytes are found. In the peripheral nervous system, they have a different type of cell, and I don't know what the cell is called. Schwann cells. Schwann cells and oligodendrocytes are the same exact thing, just where they're located. Oligodendrocytes are in the brain. Schwann cells are in the peripheral nervous system. They both provide myelin. So we've talked about the nerve, we talked about action potentials. Let's put it all together and go through an entire nerve synaptic transmission. Step 1, we have the presynaptic neurotransmitters stored in those vesicles. The action potential caused by what ion? What? Sodium. Action potential. What causes the action potential? It's sodium movement. Okay? Causes the axon to depolarize, and that wave of depolarization goes all the way down the axon until we get to the axon terminal where the calcium channels are. Calcium comes in, causes exocytosis of your vesicles. So we're going to dump out all that neurotransmitter into that synaptic, in that cleft. The neurotransmitter is going to react with a specific receptor on the postsynaptic side. We have specific receptors. What is acetylcholine always going to bind to on a postsynaptic? Well, there's 2 of them, but what's the one we learned about yesterday? Nicotinic. A nicotinic receptor is for acetylcholine. So we'll learn about, we go through the different neurotransmitters, what do they bind to, and so forth. So it binds to a specific receptor on that postsynaptic membrane. How do we stop this signal from keep going? We have to terminate it. We remove the neurotransmitter back into the presynaptic membrane, repackage it, reuse it. We can degrade it through enzymes, or most commonly, we just reuptake it. Those are the three ways we stop neurotransmitters from working. Enzymatic degradation, metabolized it. We remove it from that synaptic cleft via diffusion, or we just repackage it and take the whole thing back up. So here's a graph showing that. We have the act potential coming down that axon. We get to that terminal Booten where our calcium voltage-gated channels are. Calcium channel opens up, calcium comes in, causes movement of the synaptic vesicles to the end to be released. Once that neurotransmitter is now in that synaptic cleft, it will bind either to a ionotropic receptor, so a ligand-gated channel. A neurotransmitter can be a ligand. It's going to bind to the gate and open it up. So we learned about ligand-gated channels on the first day. Here's an example of one. You have ion-gated channels where it binds the ion and opens it, and you have these secondary messengers called metabotropic receptors. What I want you to know about these right now, all these metabotropic receptors do is they amplify the signal. That's all they do. They amplify the signal from that neurotransmitter that has a major effect on that next nerve or the next organ or tissue, whatever it's going to be. Whenever you see metabotropic receptors, G proteins is one of the big ones you may have heard before. These amplify that signal. That's the only thing they do. And we'll get to this a lot more when we get to nerves, endo, sorry. So here are some selected neurotransmitters we're going to go through. It's going to look confusing, but we're going to break it down, and most of the pictures you don't need to know. So I just put them there, but don't stress out about the pictures. So the first one we're going to talk about for neurotransmitters is acetylcholine. We talked about it a little bit yesterday. In the somatic system, what is the somatic system referring to? Body, muscles, movement, skeletal movement. We know that when this nerve signal goes down, it's going to release acetylcholine onto a nicotinic receptor. We're done with muscle. They all use the same exact thing. Always acetylcholine to nicotinic. All your skeletal muscles. So just remember that for all somatic system. For your autonomic nervous system, for the parasympathetic. Parasympathetic, is this rest and digest or fight or flight? Rest and digest. Acetylcholine is released onto a ganglion, which then releases acetylcholine again to muscarinic receptors. In the parasympathetic system, acetylcholine will bind to muscarinic. So muscarinic and nicotinic are the two receptors for acetylcholine. So what kind of receptors can you also call these? If anything that deals with acetylcholine is called a what? Cholinergic. These are cholinergic receptors. Nicotinic, muscarinic are your cholinergic receptors all the time. Later on, when we get to pharmacy and pharmacology, you're going to start learning about drugs that will cause anticholinergic effects. Because you're blocking all this acetylcholine from working, you're going to have a lot of problems downstream because the acetylcholine is not going to work as well as it should be. Someone had a question back there? Yeah. Okay. So you're blocking acetylcholine from being released with these anticholinergics. You get dry mouth, your eyes get dry, a lot of things, other things you'll learn about later. In the sympathetic system, acetylcholine is only in that pre-stuff. There are some rare instances where acetylcholine goes to a muscarinic, and it's only with sweat glands. So acetylcholine will also go to a muscarinic receptor on the sympathetic side for your sweat glands. The rest of it we'll go through as we get to the different neurotransmitters. So what do you have to know for these neurotransmitters? One, you have to know, how is it metabolized? Acetylcholine is metabolized via acetylcholinesterase. Uses, somatic and autonomic transmission, and the receptors, nicotinic, muscarinic. Those are the only two things acetylcholine will ever bind to in our body. All skeletal muscles are nicotinic, muscarinic are in their sweat glands. So that's acetylcholine. Next is norepinephrine. Also, you may see it as noradrenaline. Again, that graph, it's just there. You don't have to memorize it. What I do want you to know, again, synthesis. Here's your aged meats and cheeses. Okay. Metabolism, COMT and MAOs. So monoamine oxidase and catecholamine O-methyltransferase, just no COMT and MAO. Those are the two ways to metabolize norepinephrine, meaning we want to get rid of them. We don't want them sticking around long, so we have to metabolize them and break them up so that we can get rid of it. Function of norepinephrine, obviously it's sympathetic, so it's excitatory, and has some effect on your wakefulness. Obviously, if you get scared, you may become more alert real fast, because all that norepinephrine is floating around. The receptors, because this is mainly only sympathetic, it's going to go to your alphas and betas. There are 2 alpha receptors and there are 3 beta receptors. But regardless, anytime you see alpha, beta, it's always sympathetic. Also known as adrenergic. Adrenergic, because it comes from the adrenal glands. So you have cholinergic, acetylcholine, adrenergic receptors, alpha, beta, sympathetic. Start thinking those things always go together. Alpha, beta, sympathetic, fight or flight. Dopamine. Again, aged meats and cheeses. Okay. Metabolized, same thing as norepinephrine, COMT and MAO. There's a class of drugs called monoamine oxidase inhibitors, MAOI. So if we're going to inhibit MAO, what will stick around longer? Dopamine, which is a happy hormone. So when people are depressed, they take MAOIs, which also affects serotonin. So those things stick around longer. They're not metabolized so quickly. So they have more of an effect. Function of dopamine, a couple of things. Movement, as it relates to the nigrostriatal, again, that substantia nigra. It also has an effect on our mesocortical pathway, and that's our reward pathway. When you feel good because you do well in an exam, that's your dopamine kicking in. Because you got a reward, you feel good. That's where drug abuse comes into play. Because they want that reward, they want that feeling, but unfortunately they're going to need more dopamine to get that same effect. So they start doing riskier and riskier things, more drugs, because they want that dopamine hit. That's all they want is dopamine. And they want to find a way to get it. And so they do risky things and drink a lot and do drug use. The last one just goes to the pituitary, but the first 2 are the more important ones. Movement and the reward pathways. Receptors. For dopamine, there are 2, D1 and D2. So dopamine goes to the D receptors, easy to remember. One stimulates adenylyl cyclase, one will inhibit. So remember that fine movement, that motor control. By having dopamine with these 2 stimulators, D1 and D2, depending on which one they're stimulating, will cause an inhibition of movement or easy movement. Here's serotonin. Comes from tryptophan. Where's tryptophan? Everyone talks about it. Turkey, Thanksgiving. That's why you get tired and sleepy afterwards sometimes. Metabolized. Here's your MAO again. monoamine oxidase. So if you give an MAOI, the inhibitor, serotonin sticks around longer and you'll feel better. The role of serotonin, mood, role in anxiety, okay, nausea and vomiting, and cerebral blood flow. We give 5-HT agonists and different types of 5-HT hormone medicine. If you ever take, I'm drawing a blank on the name. No, it's for the migraines. You stab yourself with it. I can't remember what it is now. And I teach neuro too, but I'm drawing a blank. Anyway, it helps with migraines. Receptors, a lot of receptors. Serotonin is mainly made in the gut, and then it goes to the brain to make you feel better, which is why if your diet is garbage, you're going to be depressed. Because you're not eating the right foods, you're not making serotonin, you're going to feel like crap. Which is probably why a home-cooked meal makes you feel really good, because it's all natural stuff. It's like real food, and you get all the serotonin, and you feel good. Yeah, so this is basically true. That's all you want is serotonin and dopamine. And all you do every day is trying to get more, so you feel good. So GABA, gamma aminobutyric acid, just remember GABA. GABA is the major inhibitory neurotransmitter. This is the first one we learned about that is inhibitory. So, made from glutamate, requires B6, don't worry about that. Metabolism, GABA transaminase. Obviously, if I asked you, GABA transaminase does what? You know it's GABA. Don't worry about studying that. Function, inhibitory. Receptors, there are GABA channels, but this is the really important part about GABA. It opens the chloride channels. We never talked about chloride, right? We talked about sodium, we talked about potassium. We mentioned chloride, that it's there, and we know it's there, we never talked about it. Now we're going to talk about it and why it's important. Chloride, is that a negative ion or a positive ion? Negative. So if we give someone a GABA agonist, it's going to open up those chloride channels. So now chloride is going to come into the cell. It's a negative ion. What's going to happen to the resting membrane potential when all this chloride goes in? More negative. So what's the normal resting membrane potential? Negative 70. What's threshold? So we're dumping in chloride. Let's say this number now goes down to negative 90. That's where our membrane potential is now. Do you see how far away we are from 55 to get threshold? So we're already lowering so we're making it harder to get to threshold. So that's why it's inhibitory. We're inhibiting the cell from doing anything by making it more and more negative. Okay, AA people, the drugs you give are going to affect GABA because you're trying to make these people pass out. Okay, so I think the next slide talks about it. Not yet. There's a slide I mentioned all this stuff, but this is where it comes into play because you're giving them benzodiazepines. Open up these chloride channels. Okay, and we'll go through some of them more in a second. Glutamate is the major excitatory neurotransmitter of the brain. What's MSG? Monosodium glutamate. What happens if you eat MSG? It tastes great. It's like salt on crack, but what happens to you? You get jittery, you get anxious afterwards. Because it's glutamate. You're exciting your brain. You're making it more excitable. That's why they get little jitteriness, some lightheadedness. That's why people complain about MSG giving them headaches, because it's stimulatory to the brain. So glutamate can cause excitotoxicity with overstimulation. So people who have had strokes or people who have had seizures, this glutamate is going crazy because all these cells are dying. Glutamate starts going crazy, and in those cases, glutamate actually can make that zone of ischemia worse. So some of the newer drugs we're trying to give now for stroke patients and seizure patients is these type of glutamate antagonists. to shut down glutamate so we don't fry your brain tissue. That's essentially what that's doing. The important one here is NMDA is the receptor. When you start learning drugs, they name the drugs very easily sometimes, and they did it for this one. The drug that stops it is called Namenda, because it attacks the NMDA receptors. I call it Namenda. I'm not making this stuff up. Okay? So a lot of the names are easy because they just describe what they do. Lasix lasts 6 hours. That's why I call it Lasix. Oh. So that one. Also, it has these metabotropic receptors as well as the NMDA as well. So that's glutamate. Some other neurotransmitters. These ones are just, they're there. There's tons of neurotransmitters. We're not going to go through them all. We don't need to for this course. Glycine is another one. Histamine, okay, is in the hypothalamus. Effects on arousal, body temp, vascular stuff. What's an antihistamine? Well, no, antihistamine is Benadryl. You want to take it because histamine itself causes you to snot and runny nose and swelling. That's all histamine. So by taking an antihistamine, you stop your nose from running and all that stuff. But what's the side effect of Benadryl? You pass out, you want to go to sleep because of arousal. You're blocking the histamine, so it makes you feel tired and sleepy. The other thing with histamine, we talked about that hydrogen-potassium pump in the stomach and how that pumps all the acid, the H, into it. Another type of, not a proton pump inhibitor, that's the H one, but a histamine blocker, we call them H2 blockers. They block histamine in the gut because histamine causes gastric acid to go up by itself. So another way to reduce GERD is with a histamine blocker. Nitric oxide is another one. We have cannabinoids, CB1, CB2, so for cannabis, and obviously this list is not all inclusive. Okay, so here is the correlation for the GABA one. Primary inhibitory, obviously, is GABA, glycines a little bit. So your sedative hypnotic drugs, benzos, alcohols, barbiturates, the target that GABA A receptor, causing that chlorine or chloride to flux move into the cell, causing membrane, that hyperpolarization. We're going lower of that membrane. It is dose dependent. So the more you give, the more depression of the CNS they're going to have. So at first, you want to keep them in that sedated part. Obviously, here's your anesthetic part where they're totally out. And then you get, if you keep going with, keep giving them more and more stuff, medullary depression, and then coma. So you got to be very careful with these drugs because they are dose dependent. And the more you give, the bigger the CNS depression and inhibition you're going to have. So we talked before, and I mentioned that axon hillock is very important when it comes to inhibition of a signal. So we're going to talk about that right now. So effects of synaptic excitation. So we're going to make that action potential go from one nerve to another nerve. That next nerve still has to hit threshold. So the stimuli on the next nerve, on that postsynaptic side, still has to get to threshold, -55, to do things. So what we do is we have one cell body, one soma, but we have many different ways to input signals through all those many, many dendrites that they have. In order for an action potential to occur, may require multiple activations to allow you to hit threshold. So the process is called summation. And there's two different ways to summate or to add together all of these stimulatory events. One is called spatial, which is just in space. You have multiple inputs from different areas. Multiple inputs from different neurons. That is spatial. The next way is temporal. And temporal refers to time. What's the tempo? Successive discharges at a terminal. So now you just have one nerve successfully or successive discharges onto the postsynaptic side to hopefully generate action potential. So you can see here in the graph, we have millivolts here. Obviously, our resting is -70. Here's your -55, which is threshold. If you look at the red one, the red has 16 synapses firing all at once, so when you add them all together, it's enough to get to threshold, and we have an action potential. So that red line going up all the way up, that is the action potential, and we succeeded in propagating the signal. We succeeded in communicating to the next cell. But if you look at the next one, the green only has 8, and the blue only had 4. It's not enough. It doesn't get enough to get to threshold. So you can see how we need multiple inputs just to get to threshold. All of these, I'll have a better picture in a second. I'll go to the next one. Okay. So here is another way of looking at the dendrites going into the cell body. The numbers that you see are the membrane potentials and where they are. All the ones that say E are excitatory. The ones that say I are the inhibitory ones. Where are the inhibitory ones? Closer to the axon, and right on that axon hillock right there, you can see these two right here. You have some right here as well. This is a safety measure. We don't want to have unchecked excitation of our neurons that causes seizures. We want to have a way to shut them down. The best way to do that is to shut it down before it even gets to the axon. And that's why all the inhibitory neurons are going to synapse closer to the axon hillock so it can stop the action potential from getting to the axon. Once it gets there, it's going to go because it's myelinated. It's going to go all the way. We want to prevent that from happening. So by having all the inhibitory ones right at that axon hillock before it gets to the axon, we can stop any signal from propagating through. Does that make sense? Yes? Does that mean every single one of those has a trigger right there? Yeah, just for protection. But they may not be fired, only when they need to be fired. Meaning only when an inhibition needs to occur will it fire. Otherwise, it just goes. But it's just a safety mechanism to stop anything from getting out. So that's why the inhibition ones are always closest to the axon and can shut it off. If the excitatory ones were all post and there's no inhibition past it, there's no stopping that signal. It's going to keep going. So it's set up that way that excitations happen more in the distal portions and the inhibitory happens more on the proximal portions, so it stops it from getting outside of that nerve. Some special characteristics of synaptic transmission: fatigue. Obviously, there is some fatigue. Initial excitation and discharges are very rapid, but then they're going to slow and die out. It's just fatigue. They just can't be stimulated so many times in a row. They're going to fatigue out. Again, important for termination of seizures. Usually, they don't last very much, but if they do last more than 5 minutes, it's a medical emergency. You have to really break that. Again, protective mechanism, effective pH, alkalosis increases neuronal excitability, so alkalosis can cause seizures, whereas acidosis decreases excitability, and that can cause a coma. Alkalosis is where's the pH going to be? High. So pH in alkalosis greater than 7.5. Acidosis is pH less than 7.3-ish, around there. That's your acidosis. When we get to pulm, when we get to renal, we'll talk about is it metabolic or respiratory. But regardless, alkalosis will cause seizures, or it can cause seizures. Acidosis can cause a coma. I got two more slides, we're done. Some special character, another thing, effective hypoxia. So the oxygen environment. If we're in a high requirement of oxygen for neuronal excitability, so when you don't have blood flow for 3 to 7 seconds, can lead to unconsciousness because we need constant oxygen. There's really no storage of energy in the brain. It uses it all up, so we got to keep supplying it with fresh oxygen. That's why for a certain amount of time, no oxygen, you can pass out. Drug effects, caffeine, obviously adenosine antagonism, so it's excitable. And then your sedatives and anesthetics make depolarization more difficult. Okay, what time is it?
57.
Okay, yeah, we'll stop here. Let's see what yeah, we'll stop here. We'll start this up on Monday.